Hi everyone. Today we're going to be talking about steps in the scientific method and how you can look at an experiment and identify things like the independent variables, the dependent variables, and any constants that would be present in that particular experiment. So the scientific method itself is what we call systematic. That means we're following the same approach every time we're going forward and trying to test something about a particular question we have. So no matter what the science is, I'm following this systematic approach every single time. The steps are the same steps that we would follow anytime we do an experiment. The key here is that we want to be able to report results, and scientists around the world should be able to describe their methods and everyone understand what's going on. So the key here, when we're looking at an experiment, when it gets published and reported out for people, we want other scientists to be able to repeat these results multiple times over and over. If I have a study that comes out on a new type of medication, I want other scientists to be able to get the exact same results that I got to ensure that the results I, in fact, got are valid. So the key here is we want to repeat them over and over and over again. So even though you might have a great result, the first time you do an experiment, you're going to do it again and again and again. And as best as possible, repeat those results so that way they're the same across the board every time you do this experiment. Okay. So the typical steps that we would see in a scientific experiment are going to be repeated over and over and over again until you either report your hypothesis, meaning you've accepted it, it's good to go. I guess I should use a different coloring there. So it's either, you're reporting it, you're saying, yes, scientific community, it's good to go, or you're discarding it, meaning it's no good. Whatever I thought happened didn't actually happen, so I need to go back to the drawing board. So you observe observations about the world around you, and you determine that there's a question that you have, a problem. So you're going to go through and try to create a hypothesis. If I do this, then this will happen. So a lot of times you will see these written as if then statements. If I do one thing, then I will see these results. This leads us to experiments. So we're going to go through and test that hypothesis, gather data about it, and draw conclusions. If my conclusion is good to go, I'm going to move on to the theory part of the test. If it's not good to go, I'm going to go back around, revise it, and retest it again. So the reason why this isn't steps like step 1, step 2, step 3 is because you're going back through and redoing some of these depending on the results that you get. So if I get poor data the first time, I might need to go back through and revise my hypothesis and start back from the drawing board, okay? But if my conclusions are good and I'm ready to move on, that means that I now have what's called a theory, which means I have tested my hypothesis. I've tested it over and over and over and over again, and I've seen the same results every single time. So now I have a theory that whenever this occurs in nature, these are the results I get. So this constant experimentation step is really important. If I find that these supported hypotheses are something that's happening across the board in nature, this is what we would call a scientific law. A lot of times these are supported by math. So I'll put that kind of key term here. So for example, Newtonian laws of physics are explainable by math. So force equals mass times acceleration, that's a Newtonian law. That scientific law was a hypothesis that he viewed in, something about the way things work in nature. He tested it many, many times. He was able to get data over and over again that was repeatable, and he created a mathematical law to show, Newton's second law, which is force equals mass times acceleration. So as needed, we will go around and revise this theory again, kind of like what we saw over here. So the whole process, the idea of this is all repeatable. We should be able to go through and do this over and over and over again across the board. So some vocabulary terms that you'll need to know. I would not only write down these vocabulary terms on your notes, I would make flashcards as well. You're definitely going to need to know what these terms are. So the two types of data that we will see in scientific experiments would be qualitative and quantitative data. Quantitative data is numerical. So if you see the term quant, that means number. It's numerical data. So things like temperature, length, mass, etc. Qualitative data are descriptors. So if I'm looking for signs of a chemical reaction, that's not like a number that I'm measuring. It's just something I'm looking for or describing. A hypothesis then is a tentative explanation. A lot of times, again, these are written as if-then statements. And then an experiment is that it's a controlled way of observing results in testing a hypothesis. So the big thing here is it's controlled, it's repeatable, it's going to be the same every time I conduct this test. So other vocab terms you need to know, the independent variable is the thing that you're planning on changing. This is the thing that's not going to stay the same throughout the experiment. It's the thing you're modifying. The dependent variable then is what you are measuring. So this is what's getting measured as a result of the independent variable. So I change something, the results that I get, those measurements I'm making, that would be my dependent variable. It depends on what happens with the independent variable. A control is a standard for comparison. What this means is it's things I'm keeping the same. So for example, if I'm testing 10 different chemical reactions, I might want to have the temperature of the room that I'm in be the exact same, 70 degrees Fahrenheit. That would be a control. Or that I'm using deionized water for every single experiment instead of some being tap water, some being deionized water. That would be a control. So it's things that I'm keeping the same. A theory is an explanation that's been supported by many experiments. So that means I'm taking a hypothesis and I'm testing it over and over and over and over and over again, and I'm seeing the exact same thing every single time. And then a law is where we are seeing that not only is this relationship shown in nature with many experiments, but it's also being supported mathematically. All right, so different types of research that we will see in these kind of scientific method steps. Pure research is something that I am just trying to gain information about. I'm curious about something in the way that it works, so I'm going to test it out and see what happens. Applied research, on the other hand, is usually driven by industry. So I have a problem, maybe I have a drug that I want to take to market through the FDA, but I need it to be able to do something first. The scientific method steps that I'm taking to go through and test out this new medication that I'm creating is for a specific problem. So I have a specific problem. I'm trying to find an answer to that problem. That would be what we call applied research. So there is a difference between the two. Pure research is simply just I'm curious about something and I want to see what works and I want to learn more about the world around me. Applied research is usually driven by industry because we have something specific that we want to find an answer for. So let's practice identifying some variables here. We're going to go through and identify your independent variable, dependent variable, and the constants. So if skin cancer is related to ultraviolet light, then people with a high exposure to UV light will have higher frequency of skin cancer. So if we're saying that skin cancer is correlated to UV light, the independent variable would be the amount of UV light. We would expose people to different quantities. And then what we are measuring as a result would be things like frequency of skin cancer. So the amount of skin cancer depends, the dependent variable, on how much UV light is provided to that person. A constant here might be whether or not we're working with male or female people. It could be type of exposure. So it could be from the sunlight. It could be from a UV lamp. So these aren't things that are necessarily written here. I'm just kind of assuming what a constant would be, things that I would want to keep constant. So male or female, type of exposure. I might want to say their age. I might want to keep that constant. I might want to keep their, sunscreen use. Like if there's someone who wears sunscreen every single day, I might want to change that if I were conducting this experiment. So this is just an example of one. The thing that I'm changing is the amount of UV light, and then what I'm measuring as a result is how much skin cancer they end up getting. Okay, let's try another one. So in this one, if leaf color change is related to temperature, then exposing plants to low temperatures will result in changes of leaf color. So independent variable here would be the temperature I'm exposing my plants to. So temperature of exposure. The dependent variable would then be the leaf color. So here's a good example of a qualitative test, okay? I can't measure color necessarily as a number. So the color that I'm observing, you know, whether or not I'm noticing like a red change, yellow change, whatever, that would be a qualitative type of result. And then the constants I'm changing here might be amount of sunlight. I could also make sure that it's the same species of tree. I could be, let's say amount of sunlight, same species of tree, nutrition and water. So how much water and how much food am I giving these trees? Those would all be things that I would want to keep the same for this experiment to make sure those aren't impacting what the leaf color would be. The only thing I want to be exposing is the temperature of, you know, what those leaves are exposed to. Okay, so if you have any questions with any of these, please let me know. We'll practice more of them, but hopefully at this point, this is a review from classes you've taken before, and it's nothing too new or exciting.